Okay, so we just finished talking about the central dogma, and hopefully you watched the video I've posted on your Canvas shell showing an animation for DNA transcription into RNA. I'm going to briefly go over that process again and kind of highlight some of the differences and a little more details. And then we'll turn to how bacteria regulate their gene expression. Okay. So a couple things about DNA transcription into RNA. It's mediated by RNA polymerase, and that uses ribonucleoside triphosphates. And so here's an image of a transcription bubble opening here. Of course, RNA polymerase is the enzyme that opens the double helix and then begins to polymerize in the 5' to 3' direction. And it's going to form anti-parallel strands with DNA, and it's going to move in the 5' to 3' direction. And the base pairing is going to be complementary. So C is going to pair with G, but A is going to pair with U, because remember, RNA does not have thymine, instead they have uracil. And as this grows, this is an exergonic reaction because these are ribonucleoside triphosphates. And so they're attached, each nucleoside is attached to 3 phosphates. And the phosphodiester linkage cleaves these last 2 phosphates, and that provides the energy for this polymerization. So when this replication bubble forms, as it showed in the video, there are 2 strands. There is this template strand, and this is the strand that is turned into RNA. And then the other strand, we call this the non-template strand. And then I think somewhat confusingly, we also call it the coding strand because its sequence is identical, except for the thymines changing to uracils, to this RNA, this messenger RNA that is forming. So again, this template strand is the one being copied, and this other strand is the non-template strand. And you should be able to know this for our quiz and going into class. Okay, so RNA polymerase is the enzyme that adds the nucleoside or ribonucleosides to the growing RNA strand. And as I mentioned, it polymerizes in the 5' to 3' direction, just like DNA polymerase, except it does not require a primer. So recall in DNA replication, we needed an RNA primer. This does not need a primer to work. Bacteria have one RNA polymerase, but eukaryotes have several. And so that's an important difference. So RNA polymerase is shown here in green. This is also called the core enzyme. And before RNA polymerase can initiate transcription, it has to bind to this other element called sigma. This is another protein. This is shown in blue. And when these two bind together, we call this the holoenzyme, literally translated as a whole enzyme. And what sigma does is it binds to the promoter sequences. Okay, so recall that here's our DNA double helix here. And this site 1 or position 1 on the DNA template is going to be the start site for DNA transcription. But upstream of that, so this means upstream means before, upstream of that, we have some important sequences. So altogether, we have this promoter sequence. This sequence promotes gene transcription. And there are 2 important elements. So at position -10, so working backwards from +1, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, we have the -10 box. And this has some conserved sequences being TATA AT, and then 35 base pairs upstream is the -35 box, which is TTG ACA. Some bacterial promoters also contain this an additional upstream element, literally called the up element as well. So those can be in bacterial promoters. And sigma, as I mentioned, is what binds to the promoter sequence. And it binds in a very precise orientation, in one orientation, so it has to grab onto this -10 box and this -35. And so the promoter orientation determines which strand will be used for the template and also determines the direction that the RNA polymerase will move. And so that's why this promoter sequence is important, is because it gives us directionality to where we're going to start transcription. So once the promoter sequences bind to sigma, RNA polymerase can open up the double helix, and this creates a transcription bubble. And so into the active site of the enzyme, so here's our bubble right there. The template strand is threaded, so this bottom template strand is threaded through the active site. There's another site on the enzymes where these NTPs can come in, right? So they're coming in here, and they can form complementary base pairs with the DNA template. And then RNA polymerase can begin to create these phosphodiester bonds. And then the forming, as it elongates, the forming mRNA template exits through a separate port on this holoenzyme, on the RNA polymerase part of it. So that's what's going on in initiation. And so during elongation, the RNA polymerase continues to read the template. And the nucleotides are, again, being added to this 3' end, and they're just going to be added on and on and on. But how do we know when to stop? What's going to happen here? And so stopping, there's a few different mechanisms, and those are talked about in the video, for how this transcription process is stopped. And this process is called termination. The one we're going to focus on is the formation of this hairpin loop. Recall that hairpins are a secondary structure. And so there will eventually be some sequences that are inverted sequences. So this sequence will be opposite to this one. And so it's going to cause this hairpin structure to form. And this hairpin structure is going to cause the RNA polymerase to pause. And then when it pauses, it basically loses, there's some stability issues. And this causes the RNA to be released, to separate from the polymerase. And so that's the process of termination. So we're going to talk more about this in class and model it in class to get a little more detail, but that's a good introduction to get everyone started. A couple differences worth highlighting between bacteria and eukaryotes. So eukaryotes have 3 RNA polymerases, not just the one. We also have larger, more diverse promoters, including the TATA box, which if you take genetics, you'll learn a lot about. And instead of just sigma, we have a lot of general transcription factors that recognize promoters rather than the sigma proteins. And so we talked about some of these when we talked really vaguely, some of these when we talked about signaling, but also E2F was a transcription factor. At termination, a poly A signal is transcribed rather than that hairpin, and this allows an enzyme to cut downstream and release that RNA. And then, of course, transcription occurs in the nucleus, and translation occurs in the cytoplasm. So those are important differences between what you saw and then what occurs in eukaryotes. Okay. So next we're going to switch gears a little bit and talk about how this process of turning on genes is regulated. Now, of course, some genes are expressed all the time. We call this constitutive expression. So this is an important term that we throw around quite a bit. But this refers to genes that are on all the time. But most genes actually need to be turned on. And so turning them on can be turning them on at a low level or a high level, but we call this being induced. Okay, so thus control over gene expression, especially in these induced scenarios, is critical. So why is this important? So consider the thousands of bacterial species competing for space and nutrients along your intestinal wall. And the cells need to use resources efficiently. So synthesizing proteins it doesn't need leaves fewer resources to make essential proteins. So bacterial gene expression needs to be triggered by specific signals from the environment. We're going to talk about one of those examples, the lac operon, in class. So let's talk about first, though, some points of control for turning on proteins. So here's our central dogma again, and I've added a few more details. So we have DNA to mRNA to protein, but then also some proteins aren't active right away. They need an additional signal like phosphorylation to become activated. And at each step here, at each arrow here, the cell can regulate that transfer of information. And so transcriptional control, right, it means turning off a gene or turning on a gene. This is an efficient way to regulate how much of an activated protein you have around, but it's quite slow. And an example of this might be a regulatory protein affecting the binding of RNA polymerase to the promoter. And that would essentially turn off the ability to transcribe that gene. Another way is, let's say we've made the mRNA in the cytosol. I mean, this is a bacteria, so it's just in the cytosol. But we don't want it to turn into protein. So we can regulate this step as well. And this allows for more rapid changes because the mRNA is already made. And an example of this could be turning on regulatory proteins that can speed up mRNA degradation. So to turn off that messenger RNA. Okay, so this is, again, this is more rapid than transcriptional control. Finally, the last example, we have our protein made. So there's our protein, and it's being phosphorylated, or some sort of chemical modification to turn it off. So this is a way to regulate a protein that provides the most rapid response, and the activation of a chemical modification can be used to then regulate gene expression or regulate the activity of the protein. So this is really rapid. But you waste a lot of energy if you don't need this protein, of course. If you need it to be here, you waste a lot of energy in the cell, transcribing it, and then turning it into protein. So it's a balance of all these pathways. Again, we're going to focus on this transcriptional control. And we're going to look at lactose metabolism in E. coli to look at a model system for studying transcriptional control. So I want to introduce to you some important elements of lactose metabolism in E. coli. So E. coli really likes to metabolize glucose. Okay, this is a monomer. But when glucose is not present, it will metabolize lactose, which is a disaccharide. So let's talk about how E. coli is able to metabolize lactose. So the first thing that happens is that there's a membrane receptor in E. coli that's called galactosidase permease. So there's this receptor. It's made of mostly alpha helices that pass through the membrane, and it's got a binding pocket for lactose to come through. Okay, and that's depicted right here as well. So lactose is a disaccharide made of a glucose forming a glycosidic linkage with galactose, and it's brought in via this galactosidase permease. Once in the cell, an enzyme called beta-galactosidase cleaves that glycosidic linkage, and it releases glucose and galactose, and then the galactose can also be converted into glucose and metabolized. This requires more energy than just taking in glucose. That's why E. coli prefer to take in glucose. And so it's not advantageous for them to use this pathway when glucose is around. But if glucose isn't around and all they have is lactose, then this is a great pathway and they want lots of beta-galactosidase. Okay, so in this system where we need to turn on beta-galactosidase, lactose acts as an inducer. So it's actually going to turn on the genes that are needed to metabolize itself. And glucose negatively regulates the beta-galactosidase. Okay, So how? Let's talk about this. Okay, so I'll introduce you to a number of important genes, okay? The first genes I'll talk about are the ones related to lactose metabolism, and they're called LAC genes. And it turns out that they're clustered together. I'm only showing you some of them right now. So here's the E. coli chromosome. Remember, these are bacteria, so they have one circular chromosome. But very close together, there are 3 genes I'll point out. There's LAC-I, LAC-Z, and LAC-Y. We now know that LAC-I encodes for LAC-I mRNA, and that mRNA works as a repressor. So it prevents transcription of LAC-Z and LAC-Y when lactose, the inducer, is absent. Okay, so and you can think of the I for inhibition. It could be a handy way to remember it. LAC-Z is the gene that encodes for beta-galactosidase, the enzyme that cleaves glucose and galactose. And LAC-Y encodes for the membrane receptor that brings in lactose. That would be the galactosidase permease. Okay, so how do we figure out these genes? Well, we did some experiments where we made mutants. And when we made a mutant, so these were loss of function alleles, where LAC-Y was deleted, or the allele was non-functional, these cells, these E. coli cells, couldn't take in lactose. So that makes sense. That helped us determine that this was the receptor, that the channel that brought in lactose. When LAC-Z was the mutant, when it was not functional, even when lactose was present in the cell, it couldn't cleave the lactose. And that makes sense given what we know now about galactosidase. But this mutant helped us figure out this enzyme activity. And then the mutant phenotype for the LAC-I was that it made beta-gal, so it made this protein here without any lactose present. And so this suggested that LAC-Z and LAC-Y are negatively regulated by LAC-I, right? And I told you this was a repressor, but how do we know that? What is it doing? So that brings me to the LAC operon model. Okay, so I'm going to now put a few more elements on those LAC, add a few more elements to the LAC part of the LAC-Z genes I showed you before. Okay, so this is our LAC operon. This is a set of coordinated regulated bacterial genes that are transcribed together in one mRNA. So that's what I mean by an operon, and they're processed later into separate proteins. The group of genes involved in lactose metabolism is termed the LAC operon. So that's what I mean. And we talk about this LAC operon all the time because it's a great model for gene expression and how we know how a lot of genes are regulated. Okay, so here's our LAC-I gene. This is our repressor gene, and it also has its own promoter. We have our LAC-Z and LAC-Y genes. We also have LAC-A. That's part of the LAC operon, and this codes for an enzyme transacetylase that exports sugar from the cell. These genes have a promoter, so to bind an RNA polymerase, but also this regulatory sequence called a operator. Okay, so I would definitely write this down in your notes that we have a promoter, an operator, LAC-Z, LAC-Y, and LAC-A. So this is really important that you can, before coming to class, you can really know the parts of the LAC operon. Okay, so how do these elements work together to regulate glucose metabolism? So here's a cell, this is a cell where lactose is absent inside the cell, okay? So here our LAC-I gene is encoding the repressor protein. And the repressor protein binds to that operator. So when RNA polymerase, the holoenzyme, binds the promoter, it actually acts as a roadblock to RNA polymerase so that it can't move past the repressor to access those LAC-Z and LAC-Y genes and make the mRNA. When lactose is present in the cell, then the repressor turns off. So remember, lactose is our inducer. How does that happen? So, right, our repressor is produced normally, constitutively, and it binds to the operator. But lactose, it turns out, actually binds to the repressor. And when it binds to the repressor changes shape. This is called allosteric regulation. And when it changes shape, it loses affinity for the operator and it comes off, which allows the RNA polymerase holoenzyme to initiate and elongate the mRNA until it terminates. So we get production of beta-galactosidase and permease, so we can bring in more lactose and we can cleave it into useful forms. Of course, in the mutant, right, we have this instance where lactose could be present or absent. It doesn't matter because nothing is binding to the operator. So when RNA polymerase binds, it can just move straight through. So this is why we're getting that mutant phenotype that we observed. So this is how the lac operon is negatively regulated by the repressor. Lactose metabolism is also positively regulated. So upstream of the promoter, there are a few additional sequences called the CAP binding site. And the CAP protein is a positive regulator of lactose metabolism. But you can see here that it's not always bound to the CAP binding site. And so in this instance, glucose is high outside the cell. And when glucose is high outside the cell, because of signaling pathways, cyclic AMP, that secondary messenger, is actually low inside the cell. When glucose is low outside the cell, though, however, because of signaling pathways, cyclic AMP, the secondary messenger, is produced, and then the levels of that rise inside the cell, and cyclic AMP binds to the CAP protein, changes its conformation, so then CAP can then bind to that binding site, and it can aid and enhance the activity of RNA polymerase. So that's how the pathway is positively regulated. One more additional way of regulation, and this actually isn't positive regulation, is inducer exclusion. And so in this pathway, if glucose is high outside the cell, we have our repressor bound, and lactose is low inside the cell. So we're actually, the inducer, remember, is lactose. So we're making sure that no lactose is coming in when the glucose is high. When glucose is low, however, we're going to bring lactose into the cell because of the receptor, and that's going to allow the lactose to bind to the repressor, and it's going to release from the operator, and we're going to get transcription. So a little bit of a summary before coming to class: be able to describe the main steps of transcription, including the role of RNA polymerase. Recognize that bacterial and eukaryotic cells have some differences in the way they transcribe genes, and be able to name the parts of the lac operon, some of the bits about how it's regulated negatively and positively, and to recognize that it's an excellent model to study the regulation of gene expression. So that's it for my podcast, and I look forward to seeing everyone in class. Thanks, bye.